This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 494, recorded on May 18th, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. I'm all alone here in the TWiV studio in New York City, but joining me from Western Massachusetts, Alan Dove.
Good to be here. And it's a lovely, slightly cloudy, but mostly sunny day, 68 Fahrenheit, 19 Celsius, and very, very nice, which is in contrast to the storms and tornadoes we had a few days ago.
Yeah, those were something.
Yeah.
It has still been, it rained all day yesterday, all day the day before. It's cloudy now and 18 Celsius, still lingering. It's supposed to rain tomorrow. I got stuck in that storm. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's quite nice, 69 Fahrenheit, which is 20 degrees Celsius, and just kind of wispy clouds. It's been sunny, so we missed all those storms that you had, evidently.
Oh, cool.
So I was flying back on Tuesday from California. I visited Bert Semler. And in fact, during that flight, I sent Kathy to one of today's papers. She had emailed it to me. And I said, here it is from 37,000 feet. I was supposed to leave at 6 a.m. on Tuesday. The flight was delayed 4 hours. This is an interesting story. So the night before, the plane was supposed to come from L.A. I was leaving from Orange County Airport. And it was a little delayed getting into L.A., and they couldn't leave because there's a 10:30 curfew at Orange County. You cannot fly in or out after 10:30 at night. So they couldn't get the plane on Monday night. And then Tuesday morning, they didn't have a crew. They had to wait for a crew. They actually flew the plane from L.A. to Orange empty. And it arrived 4 hours late. So then we took off fine. And then they made us take a southern route. We flew southern U.S. and went up the East Coast, which is crazy, right? Usually you go straight. And the captain said, we barely have enough fuel to do this. And then as we got closer,
it's always reassuring.
He said, we have to circle here in Virginia because there's storms and the airports are getting hammered, and we'll see what happens. And then 5 minutes later, he said, we have to land. We don't have enough fuel to circle. Yeah. So we landed in Richmond, and then all the airports shut. So we all had to spend the night there.
Oh, wow.
Came the next day. So it was funny. All the passengers were lined up. It was supposed to leave at 11 o'clock. All the passengers are lined up, but there's no crew. And this guy gets up and he goes, sorry, the crew is late coming in. And everyone went, no, come on. We're here. Why can't they be here? It was the same crew as the night before. Anyway, so I got in. And the funny thing was also last part of this story, Wednesday I was hosting Ralph Barrick. Mm-hmm. Of course, he couldn't fly in. Mm-hmm. So he canceled, which was good because I didn't get home until 4 o'clock on Wednesday. So the moral of the story is only birds and bats should be flying, right, Alan?
Right. Yes, that must be good. Although the birds and bats stay on the ground, too, during all that.
And then they get predated, right?
Yeah. Yeah. In fact, in Annapolis, you could always tell when there was a thunderstorm coming because there'd be seagulls all over the grounds of the State House. They would come in and land.
Oh, take shelter.
Yeah.
That's very interesting. Very interesting. ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, June 7th to 11th in Atlanta using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial sciences in the world. Delve into your scientific niche in 8 different tracks. Don't miss this opportunity. Visit asm.org/microbe, asm.org/microbe, and use the promo code ASMPOD for $50 off registration. See you in Atlanta. We're going to be doing a TWiV in Atlanta. So come. Microbiology department at the Icahn School of Medicine is seeking faculty candidates interested in strengthening their program in virus-host interactions. Applicants must hold a PhD and or MD, have postdoctoral experience, and be interested in creating a virus-related research group that will complement pre-existing departmental programs. Recruitment of individuals or couples at the level of assistant, associate, or full professor will be considered. More information about the department and the school can be found at icon.mssm.edu. Should you wish to apply, please send your CV and a brief description of your research plan to nycvirology@gmail.com or contact Ben Tenhoover directly. Held every 3 years, the International Double-Stranded RNA Symposium is the premier conference for virologists interested in the biology of double-stranded RNA viruses. Man, we have double-stranded RNA viruses, we have plus-strand RNA viruses, we have negative-strand RNA viruses, we have probably meetings just on influenza virus. Then, of course,
meetings just on adenovirus.
And then
I was wondering if I should announce the one that's in Mexico at the end of September.
Sure, you could. And we have ASV, which covers them all. Yeah, there are lots of splinter virus groups. Anyway, getting back to double-stranded RNA virus, the 2018 symposium is going to be in Houffalize in the beautiful Ardennes region of Belgium. The conference venue is close to Brussels, which provides easy access for European and international travelers. The symposium offers scientists from around the world the opportunity to meet and hear the most up-to-date advances on a broad area of topics in double-stranded RNA virology. The organizing committee have selected an international group of dynamic speakers to present plenary talks on topics including virus structure, diversity of double-stranded RNA viruses, evolution, epidemiology, using and abusing host pathways, immunity and pathogenesis, molecular cellular virology, and applied double-stranded RNA virology. Students, postdocs, and scientists in the field are encouraged to present their recent findings. This year's program will also include a TWiV with a special guest. Early bird registration ends May 31st. There will be a link in our show notes. All right. Ebola is back.
Yes.
It was inevitable.
Well, it never really went away.
It didn't. It's lurking in the forest in some reservoir or reservoirs. And as people go in the forest to shop, to buy their bushmeat
for their groceries, yes,
they periodically catch it.
Yeah.
And the last outbreak was, 2015 in Western Africa, which was really big. I don't know, Kathy and Alan, do you remember we kind of ignored it and then someone wrote in and said, hey, you guys have to talk about it. And then we must have done it many, many times, right? Mm-hmm. Huge.
Now, wasn't there also a smaller one in Democratic Republic of Congo in 2017?
I think so, yeah. So this last one was 2017. Yeah.
Yeah, I meant to look up details because it was mentioned.
Yeah, and this thing here was the health zone of Likati near the north central border of the country. Yes, so there was one in May 2017. But of course, no one paid much attention after the big one in Western Africa. So this was first reported on May 8th through WHO, the Ministry of Health of DRC, Democratic Republic of Congo, reported 2 confirmed cases in the Bikoro Health Zone, which is pretty remote. If you look at a map, right?
Yeah, it's so this is in the kind of in the same general area as the last outbreak. The Congo River is on the northwest side of Democratic Republic of Congo. It defines, I think it's defining a border at portions of this, or no, it's not. It's pretty much running through. The Ebola River drains into the Congo River about 250 nautical miles northeast of where this current outbreak is.
So by then it had already been going on, and I guess they didn't report it. But from April 4th through May 9th, there were a total of 32 cases. There are now more. 2 of those had been confirmed, 18 probable, 12 suspected from this Bikoro health zone, and there were 18 deaths. 3 were among healthcare workers, and these people had fever, diarrhea, abdominal pain, myalgia, arthralgia, some hemorrhagic signs, and they had collected samples from them, and A couple of these were found positive by reverse transcriptase PCR for Ebola virus. So DRC has had 8 Ebola virus outbreaks in the last 40 years. And as we said, the last one, May 2017. 2.5 million people in this province with 284 registered health facilities. So the Ministry of Health of the country deployed rapid response teams. $1 million from WHO came over. Médecins Sans Frontières set up their treatment centers in this Bikoro health zone. You know, everything is going. Wellcome Trust provided £2 million sterling. And WHO says, you know, it's a remote area. It's geographically limited. You know, it's risky for the country, but probably internationally, not so much risk. But more recently, there was a case confirmed in an urban area, right?
Right. So this is in Mbandaka, which is Mbandaka, M-B-A-N. I think that's how it's pronounced.
You pronounce the M?
I think so.
I think you just say Mdaka.
Mbandaka?
Mbandaka.
Mbandaka?
I don't know.
So
but Vincent, look down
is,
in the show notes and you'll see the map.
right.
Yeah.
If you Google either of these places, you also get maps and satellite views. Bikoro is a port of sorts. It's a small town, and it's on a lake that drains into or is fed by the Congo River. And if you go upriver from that, about 70 miles or so up the Congo River, you come to Mbandaka. And that is I guess that's the big city for the area. That's they have an airport, and they're also a shipping port.
1.2 million people.
1.2 million people, and so there was a case there. So far, just one.
Yeah, I think so. But of course, and I think that's someone who visited a patient in the other zone. Right, I read that.
I think so, yeah.
I don't know where I read it, but I just remember reading that. And so that now they're more concerned because who knows if that person infected anyone in the city, right?
Right.
So
or they might have visited a funeral.
A funeral, that's right. It was a funeral, yes. Which is always, with Ebola, it's always a problem,
That's
right?
where that happens. Right, so they went to a funeral in Bikoro and then came back to Mbandaka.
Yeah.
I think so.
And so now we have 45 cases, right? And 25 deaths so far.
And they've traced epidemiological links between most of these.
Oh, yeah. What was the index? Do they know?
I don't know. So we've got this one report. There are 21 initially reported cases. 17 had epidemiological links. And I don't think they've traced it back to an index case yet.
Yeah, in the last West African outbreak, the index case was a young girl, if you remember.
Right.
But where she got it was not determined, because sometimes they can have bushmeat contact of some sort, but in that case they didn't. So they might not hear either. But that's likely where it began.
Yeah.
Some sort of contact with bushmeat.
And these areas, talking about distances, if you say a town is 70 miles from another one, that's not an hour's drive.
No.
That's these places are.
They don't have interstates.
These are not interstate highways. The way to get from place to place is you either get into a pango with an outboard and go down the Congo River, or you get on the back of a motorbike and hope that there aren't too many potholes larger than going above your head on the road going back. So it's hours and hours of travel between these two places. And apparently Bikoro either has some kind of airstrip or doesn't, or anyway, the WHO has had to fly in and out of there by helicopter, which is damnably expensive. And these are, and everything in between is jungle and kind of hilly jungle and very difficult to navigate through.
Now, what's different here is that they're going to try using the vaccine that was tested in the last outbreak. We talked about that. It was kind of introduced towards the end of the outbreak, and they didn't have a lot of opportunity. It looked like it might have worked, but really there weren't enough data to get any kind of approval. So it's, being used on a approval basis. So the country has to approve it being used, and they have. So I think 4,000 doses have arrived, and they're going to get more.
Yeah, 4,000 doses were shipped on Wednesday, and there were, I think, more being shipped.
And I think Alan was thinking of flying it over, right? From Merck.
Right. Yeah, I was just going to personally, I'll swing down to Merck and probably their North Carolina plant, and then head on over to Bicoro directly. And I'm sure that won't be any kind of a violation of international law or the rental contract for the plane or anything like that.
So I don't know if this is the point where we want to talk a little about the basics of the virus and the vaccine.
Sure.
But so to remind you, we probably did 8 to 10 episodes about this during the previous big outbreak in Western Africa. This is the country where the Ebola virus was first isolated in 1976.
Back where it all began. And Ebola virus is a negative strand RNA virus, and it's enveloped. So that membrane means it's relatively unstable to things like detergents and so forth. And it was named Ebola virus after the Ebola River rather than after the village in which it was discovered, because Peter Piot didn't want to stigmatize that village.
Right.
He wanted to stigmatize the river instead.
Yes, so now it's...
Right. And with that big outbreak in Western Africa, one of the treatments before the vaccine was giving passive immunization, so giving antibody cocktails that had been shown to be effective in laboratory studies. And then I don't know if one of you guys wants to talk a little more about the particular vaccine that's now being rolled out in this.
Yeah. So this one was developed by Merck, and it's a VSV backbone that expresses an Ebola virus glycoprotein. And I guess we have data showing that it generates a good antibody response and this has been tested in humans as far as generating an antibody response. But as Vincent said, that was toward the end of an outbreak, and so it's not clear that it really had demonstrated efficacy at that point.
So that's a vaccine that was tested in, among other places, Rocky Mountain Labs in Hamilton, Montana, which is part of the NIH. And I remember visiting there before the West African outbreak and talking with Andrea Marzi, who was the first author on the paper we're going to talk about today. And she said, yeah, we have this vaccine that looks good in non-human primates, but it'll never be tested in people because no one wants to pay for it.
Right.
And then, of course, with the outbreak, it was the vaccine, by the way, was, I think, mostly developed for the Army, for the military in case of outbreaks or bioterrorism or whatever. But it was not put into people until that outbreak. So now it's going to be deployed in what we call a ring vaccination scheme. So, if there's a case somewhere, they will identify all the contacts, healthcare workers, family, anyone who contacted that person in their daily life, and then they will be immunized. And then if there's another case, that same thing. And that's how they did the actual vaccine testing in Western Africa as well.
And this is a proven strategy that dates all the way back to the smallpox eradication campaign, at least.
Right.
Right. And of course, they don't have huge numbers of vaccines, so you can't immunize everyone. And
just the logistics. I mean, we already talked a little bit about the logistics of this place. Immunizing everybody against anything in this area is going to be a huge to-do.
And a particular challenge here, we were talking about this in the pre-show, is that the vaccine has to be stored between minus 60 and minus 80 Celsius. And that's,
that's cold.
That's what we store our viruses in the lab.
Your freezer at home is minus 20 if you have it set low enough.
And that's what we use in the lab to store viruses because it's good for long-term storage. But
so you have a minus 80 freezer in the lab that costs a fortune and uses a huge amount of electricity, and that's fine if you're running a lab. But if you're crossing mountainous jungle or bouncing along rutted roads between Mbandaka and this outbreak, that's going to be very difficult.
Plus, the electricity is not terribly reliable, according to this article.
Right. Well, that's where liquid nitrogen might work, but then you have to have a source of replenishing the liquid nitrogen.
Replenish liquid nitrogen. And I guess dry ice could work. Dry ice ethanol bath is about -80, right?
Mm-hmm. Yeah, that would work.
But again, you've got to have tanks of CO2 sitting around and keep replenishing that while you're helicoptering into this place or whatever.
Yeah, we never wanted to use dry ice when freezers went out because it could carbonate things.
Mm-hmm. Yeah.
Right.
I remember once we went on a sailing trip and we decided to use dry ice and we had carbonated bananas as a result.
That actually sounds kind of interesting.
They were good.
Yeah. Now so I couldn't get any information on the Merck vaccine storage. I mean, this is obviously a very specialized product, so they don't exactly have a standard insert for it. But looking at labs that work on VSV, this is an enveloped virus, and that's probably why it needs these super cold storage conditions. It seems like that even loses tighter at -80 after a month or two.
You know, this is unusual for a vaccine. MMR, for example, well, let's see, should be stored, maintain potency. MMR must be stored between -50 to +8 Celsius.
Right. That's much easier to do.
Yeah.
You can put that on regular ice.
And I know that in doctors' offices, you pull out the polio vaccine from a regular old freezer. Oh. So, don't know why if this is a property of, no, Kathy, you worked on VSV. You always stored it at -70, but that's just standard practice, right?
Right. And we didn't ever store it even on short term at -20. So,
yeah. So I guess any VSV-based vaccine is going to have to be kept really cold, right?
Yeah. So an ultra cold chain.
Yes, right. That's got to be a new standard, I guess, and that would be a problem even in developed countries.
Yeah. I mean, as Alan said, these are expensive freezers.
These are not installed in your local doctor's office.
No. Very interesting. It's something that has not come up.
No.
You know, we say, this is great recombinant vaccine. It works.
Right.
But we never talked about, because, the trend in vaccinology is to try and get away from the cold, right? To make formulations that don't require it. But here we are.
But the other thing is there hasn't been, I guess, the economic drive to optimize this.
Yeah, sure.
So it's entirely possible that you could have a VSV-based vaccine that doesn't have to be stored at -80, but you'd have to do that work.
Mm-hmm. Right. Yeah, there's a limited market for Ebola virus vaccines.
Yes.
As long as it's confined to Africa, I suppose. But we'll see. We're at 45 cases, and today the WHO said it's not a public health emergency of international concern.
Right.
I don't know what their limit is.
Right. So that is a term that has a very specific meaning in terms of what kinds of funding it unleashes and what policies it brings into play.
Mm-hmm.
And they met just today and just announced that it is not yet at that threshold. Yeah.
Now, they get a lot of grief for declaring these PHEICs, right? Remember for Zika, they got a lot of grief for declaring it, and even for Ebola.
I guess it costs money. I mean, it costs more money if you declare it because different things have to be put in place.
Mm.
Yes, if you declare this, then you have to commit a certain amount of resources to it. And if they've already contained this and it's about to be wrapped up and they've committed those resources to it, then they're not going to have them for the next thing that they need. And the WHO is chronically underfunded.
Yeah. But so they're declaring it today, but we don't even think the vaccine's going out till the weekend.
Right.
So.
Right. So they're saying as of today, it is not a public health emergency of international concern. They will presumably be meeting again to revisit that as news unfolds.
Mm-hmm.
So I looked up a paper entitled Stability and Inactivation of Vesicular Stomatitis Virus, a prototype rhabdovirus, published in 2013. And VSV showed a notable stability in suspension at 4 degrees C, with virus titers remaining high over several weeks. Presence of serum proteins had a stabilizing effect. whereas elevated temperatures reduce survival times. VSV dried on polystyrene, glass, or stainless steel surfaces remained infectious for at least 6 days at ambient temperature. Resistance to extreme pH, especially in the alkaline range,
Right.
but could be rapidly inactivated at 55C.
Now, I'm wondering, that kind of makes me wonder if maybe Merck is just playing it super safe with this.
Yeah.
Could be. Yeah.
Because they haven't tested it at anything except -80. And so they're going to say, all right, you got to start at -80 because that's all we've tested. And beyond that, you're on your own.
Right. And this study, of course, is on wild-type VSV.
Is on wild-type VSV, yes.
So this response, this outbreak began in April, is very quick compared to the West African one.
Oh, yes.
Where it was months before they realized it was Ebola, had already spread. So that's good in this case because they should have a better chance at containing it.
And a lot of that, I think, is because DRC has experience with this virus.
Yeah, exactly.
This is their 9th outbreak.
Yeah.
And they know what's what, and they know what's at stake. And that's not to say that things are organized and peachy in DRC. This is a place that suffered a couple hundred years of colonialism under Belgium, and then has since been in the hands of various brutal dictators. It used to be known as Zaire, and now it's DRC. But it's one of the tragic stories of Africa. It's this place that's got incredibly rich natural resources and has just been mismanaged and used as a pawn for too long.
Anyone been?
No.
Is it on your bucket list, Kathy?
Really, no.
Anywhere?
No.
Have you been anywhere in Africa?
I haven't been anywhere in Africa, and because my mother was born in Egypt, maybe I would like to go to Egypt.
Oh, yeah, that could be interesting. No, I haven't been either. Well, we will see. We'll keep you posted. Just listen to TWiV. We do this every week, and we have already done. We've talked just today longer than you will get on any news program.
That is probably true.
So stay tuned. You know where to find us.
And by the way, Bikoro is near these awesomely named places, Mutu and Local Lama.
Yeah.
I mean, this is just great. Yeah. But no, I'm probably not going to go.
You could do a TWiV from Bikoro Health Facility.
Yeah.
Or Ikoko Impenji.
That's a nice one too. Ikoko. Yeah, great names. And the Congo River is amazing looking. It's so many streams in it.
Yeah.
Mm-hmm. Anything else? That's about it.
That's pretty much the state of affairs.
That's the state of affairs. All right. Now, perhaps coincidentally, there's an Ebola paper this week that we're going to do. Just came out within the past week in Cell Reports, which is open access.
It is.
So you can check it out. It is called Recently Identified Mutations in the Ebola Virus Makona Genome Do Not Alter Pathogenicity in Animal Models. And this comes from Rocky Mountain Laboratories, which is in Hamilton, part of NIH, the Ministry of Health and Social Welfare in Liberia, and the Ministère de la Santé et de l'Hygiène Publique in Mali. First author is Andrea Marzi, who I just mentioned. And the last author is Heinz Feldman, who I had invited to ASV to talk when it was in Western Ontario a couple of years ago, and he couldn't make it. But this paper, well, let me tell you the backstory, because we talked about this story on TWiV.
With Jeremy Luban, right?
Yes, right. TWiV 415, the same 100 series that we're in now, right? So it's not too long ago. And that paper, so his group and another group had done some work looking at mutations that were selected during the outbreak in the Ebola virus outbreak in West Africa. And as you may recall, there were many genome sequences generated, which, by the way, I expect will be coming shortly from this outbreak as well.
Right.
And Jeremy's and others had focused on a particularly interesting mutation in the genome that caused an amino acid change in the viral glycoprotein, and it is a change from alanine to valine at amino acid 82, and hence we called it A82V, the famous A82V. This change was first noted early in the outbreak, I think Guinea in March 2014, and then it spread and all the isolates subsequent to that have it in it, and it was never seen before. So Jeremy's and other groups said, what's going on? So they built the glycoprotein with or without that change into VSV. So the same kind of approach that the vaccine is using, have VSV with the Ebola virus glycoprotein. And they said, if we infect cells, what's the effect of this change? And the change is that it increases infectivity of the virus by about twofold in cells from humans, non-human primates, but not cells from rodents or dogs or cats. And the change actually decreased infectivity in bat cells. And I think we did a paper subsequent from Jim Cunningham's group showing that the change increases the fusion activity of the glycoprotein. Now, the idea was that this mutation, or the theory of Luban et al., is that the mutation increases infection efficiency, so maybe it helped the virus to spread in humans. But of course, we don't know because these experiments were done in cell culture, and in
different virus.
In a different virus, of course. And I think subsequently some people did it in actual Ebola virus in cell culture also. But of course, what you would like to do is at least look in an animal model, because we can't look in people, obviously. And so that's what this paper is about. So this is Hamilton, Montana, the Rocky Mountain Labs. They have a BSL-4 facility where they can not only work with the virus, Ebola virus, but they can infect animals with it so far. So that's, and what they did in this paper was to look at a variety of clinical isolates, all right, of actual Ebola virus. And so a little background here. One thing I didn't realize, the overall case fatality rate of that West African outbreak was 40%, which is among the lowest of any so reported.
Yes.
I didn't realize that.
I didn't.
And that's the view that has emerged from this is if people actually get full-fledged good care, the case fatality rate for Ebola virus does not have to be anywhere near as high as it has been historically.
Yeah.
If you can keep them, if you can hook them up to IVs, keep them rehydrated, and manage the symptoms, this will be a self-limiting infection with an endpoint other than death. I guess that would also be self-limiting, but probably in the majority of cases, you could save these people if you had fully equipped hospitals. The problem, of course, is that you don't have fully equipped hospitals in the places where this virus tends to pop out.
Right. The reason I mention that is because very early in the outbreak, a number of groups claimed that the isolate, it's called Ebola virus Makona, is more pathogenic, it's more transmissible in human, and that's why the outbreak was so extensive. But as they say in the introduction, no convincing finding has been published that Ebola Makona bears unusual biological features explaining higher pathogenicity.
Right.
And they do mention the A82V change, which we just talked about, but of course that hasn't been studied in animals. They also mention a couple of other amino acid changes that have been studied and may confer better infectivity. One is in the RNA polymerase, and one is in the viral nucleoprotein. And I think Jeremy's and other groups studied those as well, and they do have a positive effect on infectivity. So the goal of this paper is to put these viruses into animals. And they used 2 animal models. They used immunocompromised mice, and they used non-human primates. Now, the viruses that they use here are really important. And we have a little bit of correspondence later between me and Jeremy and Heinz, and I'll tell you about that. But first, let's go through the paper. So we have Ebola virus Makona, which again has these changes, and then there's an early isolate, which would be Ebola Makona early.
Right.
It's very lyrical. Ebola Makona, right?
Ebola Makona.
So
for the rest of your days, yeah.
Ebola early lacks the changes in the glycoprotein, the RNA polymerase, and the nucleoprotein. And then later isolates, these are from Mali and Liberia, they have the changes. So that's what they're using. These are clinical isolates from people that have been, amplified in cell culture, and these are being put into animals.
And as a control, they're going to use the 1976 Ebola isolate, which has the name Mayinga, but we'll just try and call it
Mayinga.
1976.
I like that. That's a very early one, right?
Yeah, 1976.
So the first animal model, these are mice lacking the type 1 interferon receptor, IFNAR null mice. And these can be infected with wild-type Ebola virus. You don't have to adapt. So I think a wild-type mouse, you have to adapt the virus to them, and they didn't want to do that here. So they infect these mice intraperitoneally with 1,000 focus-forming units. So they have 3 Guinean isolates from early 2014, a Malian, and 5 Liberian isolates from later. So they start with a lot of different isolates. And as Kathy said, the Mayinga is the control, which is known to kill mice.
Right.
So
1976. I think it's easier just to say 1976.
1976.
Every time I hear 70, I just think of the bicentennial. Right? 1776. I do remember that. All right, so they infect these animals intraperitoneally.
Wow, you really are old.
I am old, yeah, but not that old. I'm 65.
So you remember 1976, not 1776. Okay.
Yeah. Oh, then, yeah. I don't remember 76. Kind of foggy. Oh. All right, the animals get sick, and they have a very similar disease progression. Now, the 1976 infected animals, they get really sick, and they have to sack them.
They're dead on day 5.
They just keep losing weight.
They euthanize them on
day 5. They euthanize them on day 5, but they'd have been dead really quickly.
What? Euthanasia? What about euthanasia?
No, we're still in Africa.
Oh.
So then the early and late 2014s.
Yeah, they look pretty similar.
Yeah, and they don't need to euthanize them. No. They lose some weight, and then they gain it back, and they're still alive 2 weeks later.
They do recover the growth.
And they must get a little bit of a fever or something, because in the graphical abstract,
yes, they show
those mice, they show a thermometer.
Right.
But I didn't catch any references to measuring temperatures.
So they actually feel that they have a slightly milder disease progression based mostly on weight loss in the later isolates compared to the early isolates. Okay. So the later isolates have the amino acid changes, which Luban et al. Have said, are significant for increasing something in humans, some disease or some transmission or something in humans. But here they're sort of getting the opposite, although the difference is very, very small.
Very, very small. And the viral loads in these mice are very, very similar.
Yeah, I guess the 76s are kind of slightly higher, which
yes,
is statistically significant. But yeah, the Makona early and later are pretty similar. Yeah. They do a series of inoculations from 100 to 100,000 focus forming units. And again, they get similar results at each inoculation. The 1976 is quite lethal as you increase the dose. But again, the later strains, they tend to see weight loss and severity is slightly less compared to the earlier isolates.
And that's regardless of that A82.
Yeah.
Right.
So that's the mouse model. Then they go into macaques. These are immunocompetent macaques, which I like the way they say the ultimate animal model for filovirus pathogenesis, and the most expensive as well.
Yes.
These are expensive animals, and then the BSL-4 is very expensive to maintain.
Yeah.
Boy, these are expensive experiments. So they infect these with fewer isolates, all right, because they've sort of gotten a hint from the mouse experiments. They don't need to use all of them. And that's, of course, also to save money because they're very expensive. Yeah. And they infect them. So you have to be a little more judicious. In mice, you can use a lot of mice and check a lot of different strains. But with the macaques, I don't know what you pay for a macaque, but, many thousands of dollars per animal. So you should be judicious. And they infect these animals intramuscularly with 1,000 focus-forming units. And again, the 1976 infected animals, they have to be euthanized by day 8. There's an endpoint that you have in your protocol. If this happens, we will euthanize the animals.
Right.
Now, what did they compare in these animals? Lots of things. Serum cytokines, clotting parameters, because the virus infection interferes with clotting, fibrinogen, thrombin, protein C, platelet counts, blood chemistry like blood urea, nitrogen, liver enzymes.
And again, because of the cost of these, you want to get absolutely as much data as you can possibly find out of each animal.
Yeah.
So they do.
And again, early, late behave similarly.
Yeah.
Endpoint organ titers are very similar histologic lesions. Now, the 1976 infected animals had a little bit more severe pathogenesis in terms of, necrotic cells and so forth. But the early and late, the lesions in the different Makona isolates were, they say, essentially indistinguishable. Now, one thing they do mention here, they sequenced virus recovered from tissues of the animals to make sure that there were no changes at these glycoprotein, nucleoprotein, and RNA polymerase sites, and they say there's no reversion or change there, which is good. So again, all the Ebola Makona isolates caused lethal infections, slightly reduced pathogenesis late compared to early, and of course, less than Myinga. And then the last set of experiments, these changes may, or the theory is they might affect transmission of virus.
Right.
So they say, let's see if these viruses are shed differently from the animals. So they swabbed animals at different times after infection, mucosal membranes, oral, nasal, rectal, and urogenital mucosa. They quantify virus by RT-PCR. And they say they do not see any significantly differences in shedding among the different isolates, and particularly the early and late Makona isolates. So there's no shedding differences anyway. And, go ahead.
I was just going to say, and if you go back to when they were looking at virus titers in various things, for the later Makona virus infections, there was really low virus yield in the urine.
Mm.
They don't say that it's significant, but it's kind of hard to believe that it's not. But in any case, that would also seem to imply not shedding from urine.
Yeah. And then the last set of experiments, they go back to cell culture. They say, is there any, because Jeremy's work was all in cell culture, right? So they say, okay, let's look in cells to see if we're missing something here. Now we should say that this is Ebola virus and Jeremy was VSV with Ebola virus glycoprotein. So it is very different. So they infect Vero cells, African green monkey kidney, human liver cells, HUH, and with all these isolates, including Myinga. And again, in the human cells, the early isolates grew to higher titers than the later isolates. These are the Makona ones again. So that's sort of similar to what they're seeing in the animal models. So the early isolates lack these mutations. They grow better than the later isolates. And they say they differ from the previously published data. So, of course, they're very different because these are Ebola viruses and so forth.
Right.
But what would be implied by Jeremy is not seen here. We'll get to Jeremy's thought of this in a moment. So that's the overall picture here. The early and late Makona isolates of Ebola virus from the West African isolate, which have particular amino acid changes that have an effect in cell culture and pseudotyped VSV viruses, they don't significantly affect pathogenesis or shedding in these experiments here. And they point out that this seems to differ from the in vitro previous cell culture experiments. Now, they make a point that I want to just touch on because it's really interesting. And this has to do with the evolution of a virus as it passes through a population. And I read from the discussion, as a pathogenic virus passes through the host, attenuation to a less virulent phenotype may be an alternative scenario to adaptation. And I like that very much because, the assumption is you get this mutation selected for, it's got to make it more transmissible or more virulent, but it could also...
Might just be attenuated.
Could attenuate it because maybe you want to have a balance between transmission and virulence, right?
Right.
In fact, they talk about that, there may be a need to have a balance between infection and transmission.
And classically, a way to attenuate a virus is to repeatedly pass it through a non-natural host.
That's right. So they say our data support attenuation rather than adaptation of Makona during the epidemic. And that sort of makes sense with the case fatality ratio, although as Alan pointed out, that's probably in large part to better care, right?
Right.
And not the virus itself. But they say although not the objective of our work, it seems we support the previous observation that Makona is less virulent than Mayinga, which is 1976. And I like this final thought of theirs. They say it might be time to revisit non-pathogen related factors when trying to find explanations for the unique characteristic of the West African outbreak, like poor healthcare systems, mobility of the population, higher survival rates of patients, virus persistence, and convalescence. Those are the things we don't often think about or talk about. It's harder to deal with those, right?
That's why we don't talk about them.
It's easier to say, there's a mutation in the genome.
Yes, it's easier to say it's a mutation in the virus than to say that the country is falling apart. Yeah.
And just because it might have sounded counterintuitive, higher survival rates and virus persistence in convalescence could give more possibilities for the virus to be transmitted, potentially.
Yes.
Exactly.
That's important.
Now, they conclude that we did not find any evidence that the Makona changes in the Makona increase transmission pathogenicity. And they say, of course, animal model is a surrogate. It's a proxy. But that's also the case for cell culture work, which was the case in the other story. So that's the story. Now, I originally, I invited Jeremy to join us. I thought he might like to discuss this, right? But he said he couldn't, but he said, I have a concern. And he said, me and a number of people don't think the conclusions are justified of this paper, which we've just mentioned. He says, my main critique is that the Mayinga virus was not well defined.
That's the 1976 virus.
Yeah, and almost certainly possesses a lab adaptation in the glycoprotein that renders it hyperfusogenic. Even if it did not, it's like comparing apples and oranges. But Mayinga is not really the focus of this paper, right?
Right. They're comparing the early
Mayinga
and the late.
Makona, yeah.
Makona, yeah.
So, okay, what about Makona? And then Jeremy says, well, with Jens Kuhn, we are generating, he says, the proper cloned ancestral Makona background on which to test the effect of the mutations. This is the experiment that needs to be done, and probably the only experiment that will be able to say anything conclusive regarding the mutations.
So I guess he means a Makona background from before 2014?
I think an early Makona, right?
Right, but that was done in this paper.
Right.
Yeah.
That's what was used.
Well, so Jeremy says proper cloned, which means he wants to recover it from cloned DNA, right? But I would argue that clinical isolates are the way to do this initially, right?
The clinical isolate is the real deal.
Yeah. So anyway, I sent Jeremy's comments to Heinz. I didn't say it was Jeremy, but I said, I heard that this Mayinga has this problem. So Heinz said, the Mayinga we got from CDC, which is the source of all Mayinga viruses in the country, worldwide likely. The virus we use came through Winnipeg, has 5 passages in tissue culture, virocells, and he cites a passage from the paper that mentions that. This is the virus that has been studied extensively in vivo. Most of the early molecular in vitro work was done with an 8A variant of Mayinga. The one we use here is 7A. I don't know what those numbers mean. I believe our stock is as defined as any stock. I cannot comment on hyperfusogenicity, but I would assume if it exists, it would be same or similar for the Kikwit.
Okay.
I don't know. Yeah, I don't know why they're arguing about Mayinga. It's not the point. Nevertheless, this is not the topic of the paper, right? Which is the comparison of the early versus late Makona isolates that all have the same passage history.
Right.
I think the issue between the in vitro previous papers and in vivo our paper is the use of clones versus clinical isolates. People likely have different opinions on this topic, but for me, I would see the benefit of clones for mechanistic work, but not necessarily for questions on infectivity, pathogenicity, and transmissibility in a host population. These are claims that are difficult to make for studying clonal viruses in tissue culture. Of course, animal modeling is also only a surrogate, which we state in our paper. Mm-hmm. I think is a good point, because if you have multiple mutations, you don't want to have them singly. I mean, I guess in the end, you might want to isolate them to see their contribution, but in the virus that's in people, they're all together.
Right.
And so maybe that's why the results are different, right? Because the late Makona had all three mutations and probably others as well, right? Which could be acting together, and that's why the experiment is different. So if Jeremy just puts A82V into early Makona, I'm not sure that that's the right experiment.
Yeah, it's reductionist.
I mean, you could do it. I mean, I would want one with all three, right? So
right.
Well, and it depends on where Jeremy plans to take it from there. Because if you're putting those things in, and you're going to do, as Heinz mentions, if you're doing mechanistic studies, then you want to standardize on a virus and say, okay, this is what we're going to do our mechanistic studies on.
Yeah, sure.
And then stick with that. But if you want to draw any conclusions about pathogenicity or transmissibility, you want to be as close to clinical isolates as you can, I think.
Yeah. Anyway, I think that these conclusions are, the Feldman paper, the conclusions are justified, because they're really, if you compare the early and late Makona, they're very similar. And the changes are not really significantly impacting pathogenesis in either mice or monkeys.
Yeah, I buy it.
I'll be interested to see, what Jeremy's experiments in animals tell us. Well, I'm sure we'll talk about that one as well. So, I mean, if I were to say what's going on, well, I would say, well, these changes were selected for during the outbreak. Maybe they attenuated, and sometimes that happens. If you look at the infection of rabbits in Australia with the myxoma virus designed to eliminate the rabbits,
right? Over
time, the virus evolved, as well as the host, to become less virulent. So that could have happened in this outbreak with such extensive spread. And, I always worried about the twofold effect of these changes in cell culture. So that could go either way, right? Anyway, so we'll see. It's an interesting, and it's kind of neat that the outbreak and this paper came out at the same time. Well, it's not neat, but
yeah,
it's useful to be able to talk about both.
Yes.
Yeah. We had a seminar speaker here yesterday, Erica Ullman-Sapphire from Scripps Research Institute. She gave the G. Robert Greenberg lecture in biological chemistry, and she is someone who's worked a lot on Ebola virus structural biology. And I was not aware really of this big consortium that had been put together, where they had, I can't remember now, maybe I have it, 40 labs from 5 continents contributed 200 immunoglobulins to Ebola viruses, and they blinded them and then shipped out these 200 immunoglobulins for labs to test. And they measured 38 different features. And one of the big take-home messages was that neutralization does not correlate with protection. And if you use neutralization as your funnel in different assays, that at the end you may have eliminated the protective antibodies. And so it's just a really cool story. She had a paper in Nature back in early 2017 talking about how to turn competitors into collaborators. And so the individual investigators can still work on their own antibodies, but they don't, in this 200 antibody array, they're blinded. And so everybody's contributing and can't be championing their own antibody. So, but the cool thing is they're making really nice progress in identifying features on the glycoprotein that are important for antibody interactions, particularly the protective antibodies. And so that's going to be useful both in the passive immunization kind of strategy, but also potentially in designing or engineering the best antigens for making good vaccines.
Right.
So,
Did she have any comments about the current outbreak?
she just mentioned it really briefly in passing at the beginning. So, yeah.
We had her on TWiV 2 years ago.
Mm-hmm.
Alan and I in Boston, remember?
Yes.
Cards in a hand, TWiV
She was great.
394.
Yeah.
All right, let's...
And wait, was she the one who the Girl Scouts named
Yeah. Yes.
coolest? Yeah.
Coolest something, right?
Yeah.
Yeah. Did I put a link to that? Let's see in the show notes. I might have found it. Nope. Yeah, she mentions it. Something about Girl Scouts.
Yeah.
We could probably Google it. Coolest mom or something.
She had a really cool pointer, I'll tell you that. I have not seen this before. None of us had. When she would use the pointer, all of the slide would turn gray except for where she was pointing to, and that would stay highlighted and bright.
Wow.
So it was this big circle.
That's
cool. Cool women of 2016.
Cool women of 2016. All right.
Yeah.
Yeah.
All right, let's do a few emails. We have Anthony writing about this New York Times article that we mentioned, I don't know, last time. Trillions upon trillions of viruses fall from the sky every day. He writes, I believe there was a remark, something like, it's not as if there are cells up there, but there are. And he sends a link to an article. Bird, plane, bacteria, microbes thrive in storm clouds. I don't think we said there weren't cells, but we said there,
Yeah.
In fact, I think we mentioned that we knew that things like fungal spores could be carried this way.
Yeah. I might have thought that maybe there aren't cells up there. I don't know. But this story that is linked to is an NPR story that mentions skyhooks, which is the way that you can sample air from an airplane. And it links to a paper by someone named Meyer, who uses notes from Charles Lindbergh. And Lindbergh sampled the air using these skyhooks. And so there's notes of Lindbergh's. It's just an amazing paper. It's really cool. And they have pictures of all the organisms that they found and stuff. So they clearly look like cells and spores and things.
He continues, I've wondered if clouds could carry influenza from migrating birds flying over or through them. A dark cloud preventing the passage of UV could be the silver lining for the virus. There was also mention of much material originating from the ocean. That's not water from a glacial spring mixed with sterile sea salt. That's excrement from marine mammals, waterfowl, raw sewage, and who knows what else. I'm thinking of the DNA fishing expeditions finding sequences coding for unknown proteins. Vibrio and clouds might not fall mainly on the plain, but it could find its way there. In Sarasota, Florida, one August some years back, I wondered if the hot, humid air is as alive with microscopic life as a coral reef is with things immediately visible. Yes, viruses in space would need to be inside something to shield them from radiation. If that is so, entry into the atmosphere is not impossible. Then he sends a link to a article where, you remember STS-107, the space shuttle that blew up when it re-entered, they happened to have some C. elegans experiments on that, and they survived. Apparently they were in these canisters, and they found them, when they were looking for pieces.
Yeah.
And they were alive in 4 out of 5 recovered canisters. I don't know what these canisters were like, but, I would think on re-entry they'd burn up, but they didn't. Anyway,
did Astro Kate talk about that? Somehow I remember hearing about that.
Maybe she might have. I don't remember. Are viruses likely to arrive from space today or tomorrow? Probably not. Over 5 billion years in the casino of deep time. Low probability is a winning hand.
I love that. I love that.
So this article in the Times, it says maybe viruses arrive from outer space, and but we have plenty of good evidence that they also evolved here with life.
Yes.
We have a nice sequence, so you don't need to impose that. And I think things coming from far away, it's hard because there's a lot of radiation, right?
Yeah.
I'm just not sure if that would work, but who knows? That's the kind of thing you can't get a lot of evidence for. But we do have good evidence for evolution of life right here on Earth.
Well, we need to do a really long-term experiment and shoot some capsules of various life forms towards these exoplanets that we're discovering. And then several thousand years from now, somebody will be able to go there and see what happened.
Anthony also sent an article by Laurie Garrett, Why Trump's New CDC Director is an Abysmal Choice.
Surprise, surprise.
I'm trying to find the thing that I found most disturbing. It'll take me a minute.
Go ahead.
But
so I met Laurie Garrett at the Science March here in New York City a couple of weeks ago. And this is a good article. It's very detailed and well thought out, and why. This is a person who used to work at the University of Maryland in College Park, which is where we're going for ASV, right?
ASV, yeah.
Right. I found what it was. In the early 1990s, Redfield, who is this person heading the CDC nominee, he backed a House bill that would have subjected people with HIV to testing, loss of professional licenses, and would have effectively quarantined them. Luckily, that bill died in Congress, but he was in favor of that. And he was an advocate of the so-called ABCs of AIDS, pressing to prevent HIV infection through sexual abstinence, monogamy, and the use of condoms only as a last resort.
Where do these people come from? I don't get it.
They crawl up out of the swamp.
Oh, my gosh. Anyway, he was paid a lot of money and was going to be paid a lot of money at CDC, and apparently they reduced it. He agreed to work for $200,000.
Oh.
Nice. Good for
poverty
him.
level.
All right, we have one more from Neva. Kathy, can you take that one?
Neva writes, okay. Thanks, Twivissimos, for your well wishes for my recovery from the broken ankle. I very much plan to be ambulatory, if not dancing, for the 500th TWiV in Austin. I so much admire and appreciate the whole crew from all the TWiV shows for your professional, collegial, and very entertaining podcasts that lead the way in science communication. I find these pods and others in science and technology very refreshing refuges from the political goon show, as well as just being flat out fascinating. And Rich, your offer of schlepping me up to Austin for the 500th is more than kind, but I should be zooming, wobbling by then. Best to all and onward. Neva from Buda.
Cool. We look forward to seeing you, Neva.
Yeah. Pretty cool. All right, let's wrap up this Ebola-centric show with some picks of the week. Alan Dove.
I would like to wish a happy 35th birthday to PCR.
Wow. 35 years.
My pick is an article that I did, one of these tech features for Science, on the 35th birthday of PCR. And when I got this assignment, they said, we want to do something for the... I said, wow, 35 years of PCR. Wow.
Yeah.
How it has changed science and everything, right?
Yeah.
Oh, gosh. I mean...
So it was May of 1983 that Carrie Mullis walked into a lab at the Cetus Corporation in Emeryville, California, and performed the first PCR reactions.
I like how they say he has since moved away from the scientific mainstream. Yeah, he's surfing most of the time.
Yes, that was...
They being Alan Dove.
They being me, yes.
When did the publication come out then?
Which one? Oh, the PCR publication? So the first paper, I remember looking at this because we were trying to figure out which thing we should peg it to in determining when we should publish. The paper was 84 or something. I've got it probably in my notes folder.
Okay, because there's two references to your story, but yeah.
Yeah, so the first paper was that was the PCR amplification of the sickle cell trait gene, and I loop back to that at the very end of the article as well.
You know, we all know that it's revolutionized research, but just think of so many other things.
Yeah,
in our podcast, the epidemiology, the diagnosis that you can do we could never do before. And of course, it has led to the eternal question, does nucleic acid means there's infectious
Right.
virus there. Yep. Do you guys know that Carl Zimmer and The Atlantic ran some articles this week? Because some very old hepatitis B sequences were pulled out of very old graves. Mm-hmm. And, the articles say, oldest virus isolated. And of course, they haven't isolated any virus, but.
Right.
Sorry, Kathy, go ahead.
No, I was just going to say, back on the PCR story, I remember we had a journal club when I was at UGA with Lois Miller's lab. And I remember Lois Miller presenting the first paper about the polymerase chain reaction. And because we didn't always all read the papers before the journal club, let's say I didn't always, it was a little hard for me to understand, because the whole idea was that you could amplify DNA that you didn't know the sequence of, but you had to use primers that seemed to be specific for the DNA that you didn't know the sequence of. So I just remember that being the most counterintuitive thing and being kind of confused by it.
Right.
And of course, it became much clearer as time went on. And so I kind of think about that when there's complicated new technologies that I can't quite get all the nuances of. I just figure, eventually I'll get it when it's more textbook-like or something.
That's how I felt with the CRISPR diagnostics papers that we did a couple of weeks ago. I was having a lot of trouble with some of them.
No, that's exactly where I was thinking about it too. Yeah.
Kathy, what do you have?
I picked something that's the title is Everything Can Be a Speaker. And it's this really cool story that if you take high-speed video of what looks to be an inanimate, non-moving object, when there's sound, either music or speaking going on nearby, the high-speed video can pick up the vibrations from the sound, and that can be sort of not calculated is not the word, reverse engineered to give you the sound again. So they show a plant that can end up being a speaker. They film some Apple earbuds that are playing a song, and the Apple earbuds vibrate enough that the camera can pick it up and recover the audio. And then they were able to put that through Shazam, and Shazam could identify the song. So it's just really cool. A bag of chips, a soda can, a potted plant. The best material was a thin foil wrapper from a Lindt chocolate bar to be that. But you can imagine now this might show up in CSI type of shows or mystery novels or things like that. I thought it was pretty cool.
Or Google may start mining your videos for incidental stuff that they can then sell to advertisers.
Right.
It's great. It's pretty amazing. I have two article picks. One is an article on The Hill, which is
yes,
so sad. A Republican lawmaker says that rocks falling into the ocean are to blame for rising sea levels. Now, this person is a representative from a Republican representative from Alabama. He's a member of the House Science, Space, and Technology Committee. And he said this at a hearing on climate change. And he said rocks, the rate of global sea level rise has accelerated. And it's because rocks and soil falling into the sea have done that.
Yeah, there's more erosion now. And so
we
have more of that happening.
So on Twitter, Craig Hockenberry did a calculation, a back of the envelope calculation. It's called "Let's Throw a Little Math at Mo Brooks Rocks," and you need to put a lot of rocks. The area of all land on Earth is 57 million square miles. If you scraped a half a foot of Earth off all land, you'd get the 2.6 inch displacement. That's a lot of rocks.
Yes.
Okay, so that's one. And the second is a Washington Post article, which just came out today, where Bill Gates said multiple times that Donald Trump has asked them, what's the difference between HIV and HPV? And I find that very sad that these are two viruses that are really in the news. And not to know this is really inexcusable. But more scary maybe is later in the article, Trump asked Gates if vaccines weren't a bad thing because he was considering a commission to look into the ill effects of vaccines. And Gates said, no, that's a dead end. That would be a bad thing. Don't do that, which is great. Thank you for supporting vaccines, Bill Gates. So there you have science at the highest levels in our government. And we have a listener pick from Noah, who sends a link to an introductory virology lecture by David Baltimore on iBiology, which is a company that makes all these videos. Introduction to Viruses. Baltimore outlines the sequence of events that led to the Nobel Prize-winning discovery of reverse transcriptase. iBiology, if you haven't seen them, they put the speaker in front of a slide, and the way they record it is funny. So you don't actually point at what you end up pointing at, and it looks a little odd, but some of these are pretty good. If you want,
it's like the blue screen weather projector.
Pretty much, yeah.
Right.
They put it on behind. Yeah. I think you look at a monitor in front of you, and you kind of move your hand to sort of be in the right area, and then they stick it behind it afterwards.
Green screen is tricky.
Yes. Anyway, if you want more lectures, my 2018 virology course just ended, and all the lectures are on YouTube. 24 lectures this year, so check them out. And that'll do it for TWiV 494. You can find it at microbe.tv/TWiV. That's where you'll find all the show notes. So if there's a link to something you want to hear about, when you finish driving or when you stop exercising, you could go to the show notes and find that. But usually you'll listen on your mobile device on a podcatcher program. Please subscribe. Search for TWiV, subscribe so you get every episode. It's free. It costs nothing for you to download them. So please do that. And if you'd like to send questions or comments, the email is twiv@microbe.tv. If you'd like to send some financial support our way, you can go to microbe.tv/contribute. There are a few ways you can do that. Easiest, just give us $1 a month. It would really help out. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.